Hello and welcome to Science Research Weekly, Season 4, Episode 22, Pandora's Post-Bayesian Box. Get ready for Pandora's box of respiratory pathogens, mixed up earthworm genomes, the insane energies of extreme nuclear transits, a virus-like clade of Archaea, R version 4.5.1, codename Great Square Root, the new declarative physical modeling language Dyad, Post-Bayesianism using R, and the NIH Director's Transformative Research Awards. This episode covers material from June 13th to June 19th. Let's get started with some research articles. First on the docket is antibacterial Siderophores of Pandoraea pathogens and their impact on the diseased lung microbiota. So respiratory pathogenic bacteria from the genus Pandoraea turns out they can deplete iron from other microbes in the lung, leading to a rise of their pathogenic activity. So researchers actually discovered this trait from genome mining. They found genes for a siderophore involved in the process. Now, Pandoraea bacteria are especially dangerous for cystic fibrosis patients. So learning more about how they starve helpful bacteria in the lungs may lead to better treatments against the antibiotic-resistant Pandoraea. Moving along, let's talk about towering behavior and collective dispersal in Caenorhabditis nematodes. So C. elegans, model organism for nematodes. Nematodes can swarm together to create structures which are called towers. But now researchers have observed this behavior in the wild. Previously, it was only seen in artificial environments like the lab. They'll be able to actually see this in fruit orchids with rotting fruit. The nematodes gather together, they sort of swarm together to form a towering structure. I mean, at least from their scale, that acts as a sort of distress behavior mechanism in adverse conditions and allows them to disperse to better environments by reaching a vector. The vector, I think in this example, in this paper, was like a fly lands on it, they're able to disperse onto the fly leg and get to a different environment. So kind of very interesting. You don't think of microscopic worms as creating towering structures, but apparently they do, and they do it in nature, not just prompted in the lab. All right, let's turn our attention to the stars, or at least the cosmos, with the article An Episodic Burst of Massive Genomic Rearrangements and the Origin of Non-Marine Annelids. I jumped the gun there. Actually, we're sticking with worms. We'll look at the stars in a moment. So, higher order taxonomic relationships among annelids, so that is segmented worms, it's really tricky. And I should know, I'm investigating them for a part of a larger taxonomic side project right now. And so as it turns out, the story is even more complicated. By using the full genome, rather than just sort of marker genes, a full genome of species of terrestrial annelids, so like earthworms, it appears that annelids were able to totally rearrange their genomes in just a very brief period, meaning that the 2D and 3D genomic structure and organization of land-dwelling annelids is very different from their marine brethren, the polychaetes. So it's really wild that they were able to do this. It adds evidence towards one of the theories in evolution called punctuated equilibrium, where large changes can happen over a very short period of time. Okay, now here's the cosmological article I promised you. The most energetic transients. Tidal disruptions of high mass stars. So these cosmological objects rival the energy of supernovas. Their ENTs, or extreme nuclear transits. It happens when a massive star passes close to a supermassive black hole and accretes matter. Now, while very rare, they have been detected, including the most energetic transient event recorded to date, Gaia 18CDJ. Okay, Turning to our next article, estimates of submicron particulate matter, PM1, concentrations from 1998 to 2022 across the contiguous USA, leveraging measurements of PM1 with nationwide PM2.5 component data. So fine particulate matter, abbreviated as PM2.5, is a known atmospheric health risk. However, smaller particles like submicron particulate matter, that PM1, also likely to pose health risks, but they're harder to measure because they're smaller. Now, however, researchers in the US have been able to map these on the nationwide level for the first time, utilizing data, as the title says, from 1992 to 2022. 1998 to 2022. Okay, turn our attention to our next article. Landscape explicit phylogeography illuminates the ecographic radiation of early Archaea reptiles. So researchers were able to model the phylogeographic dispersal of Archaeosauromorphs. That's the group that arrives at dinosaurs, including hints at movement through even the most inhospitable climates. Apparently, there's a Pangaean tropical dead zone, so after the Permian extinction event. So kind of very interesting here of how dinosaurs, or at least their ancestors, radiated through the land masses. All right, closing out, we have a cellular entity retaining only its replicative core, hidden archaeal lineages with ultra-reduced genome. So Archaea continue to surprise. There's a new Archaean species, Caenorhabditis mirabile. It's been described as the smallest known archaeogenome, no metabolic pathways, only a replicative core. So it really blurs the line between cells and viruses. Last week, we talked about giant marine viruses, which sort of like are more cell-like than most other viruses. Now we have Archaea, these Archaea that are more virus-like than other cells. So very interesting exploration there. All right, let's turn our attention quick to some research tools. R 4.5.1 was released, codename Great Square Root. On the Python side, the 2024 PSF Annual Impact Report is now available, including that Python is the most popular language on GitHub. And in a new article on Julia Hub, there's one about something called Dyad. It's a new language for hardware engineering to make it as fast as software. So apparently Dyad is a declarative physical modeling language. So it might be worth keeping an eye out on if you're a hardware engineer. Now on the SAS side, from Stu, he has a recent blog called Boost ML Accuracy with Hyperparameter Tuning and a fun twist. It's all about tweaking the parameters of our machine learning models the right way to get more improvement out of them with a cake baking metaphor or analogy throughout. On the R bloggers side, 2 new posts. First from R on Every Day of a School Day, creating a question bank using Google Sheet, Plumber, and DigitalOcean Droplet. So the point is to, quote, learn how to build flashcard style question bank using Google Sheets as storage, R's Plumber API, and hosted on DigitalOcean Droplet. Step-by-step setup, deployment, and tips. So even if flashcards are not your thing, this seems like a really generalizable approach to do a lot of things. Even some of my back burner project ideas like an etymology database. So I'm definitely digging more into this methodology. All right, second, from Richard in Statistics, R, Post-Bayesianism. Let's try it. So what is Post-Bayesianism? Well, that's what the article is about. So I'm dipping my toes in this as well. That's kind of an interesting framework shift that I want to learn more about. All right, 4 new CRAN packages that might be of interest. There's GEESS, Modified Generalized Estimating Equations for Small Sample Data. G-Series, improve the coherence of your time series data. SS-Util, sample size calculation tools. And TidySpec, spectroscopy analysis using the Tidy data philosophy. And closing our time here, 3 new grants to look at, especially now that the NIH grants are rolling again to some degree. First, from the DoD, Department of Defense, some breast cancer grants, including ones of Breakthrough Award Level 1 and 2, which supports promising research that has high potential to make breakthroughs in breast cancer. There's one from the NIH, the Support for Research Excellence First Independent Research, or SureFirst. It's an award. It's an R16, clinical trial not allowed. And it's under a bunch of different categories you can kind of look at on the page. And it supports research capacity building at eligible higher education institutions through funding investigator-initiated biomedical research in all sorts of fields under the missions of the NIH. And then third from the NIH as well, the NIH Director's Transformative Research Awards. These are R1 clinical trials optional. And they support individual scientists or groups of scientists with bold, groundbreaking, exceptionally innovative, original, or unconventional research with the potential to create new scientific paradigms, establish new improved clinical approaches, or develop transformative technologies. Cell virology, thanks for joining me for Science Research Weekly. Science on